Yeah, so we're going to start out by defining some of these terms based on this little graph here. This little graph is what we're going to work with. That's the graph of an action potential. And so depolarization is a potential that's going to move in the less negative direction. Okay. Hyperpolarization is going to be when we're going below or more negative than resting membrane potential. And then repolarization is moving more negative towards resting potential. Okay, so just take a sec there and kind of just, yeah, label each of those with the letters and then decide which one answers the clicker question there on the right. Do that, just check with the people on your team, make sure you're on the same page. All right, so check with the people on your team. You're going to send in the answer to that clicker question there. All right, give you about 15 more seconds to go ahead and send that in. All right. Anybody else? Might be about it. I'll leave it open for just a sec. So about 3/4 of you are saying C. Okay, so C would be definitely hyperpolarization. So C right there. What would we call A? Yep, that's depolarization, and then repolarization is B. So those are pretty much the three main phases of the action potential. And we're going to look at what causes each of those today. Okay, questions about that at all before we keep going? Okay. All right, let's go ahead and just answer this review question here. Let me start the question over. So this is pretty much what we talked about last time. What is it that is considered electrical current when we're talking about a cell membrane? Double check with the people on your team.
If I write it out in here, is that like?
Have you checked your points on there? You might already be at like 38 out of 40 or something.
32 on 24. Wait, is that what it is?
It's going to be 40 eventually. So that means you only need 3 more times to actually, but you probably have, because it's 2 points per. Yeah, you know what about. Yeah, I put the points in before each exam, so I'll put them in again before the next exam, as long as you've done 3 times using it.
Yeah.
That'll be the 6, or the, maybe 4 times, and that'll be the 8 points you need.
Okay.
So should be fine. Yeah.
Yeah, no, that's valid. It's not like, hey, My dad.
All right, go ahead and send that in if you haven't. I'll just show you what it looks like so far. So why don't you guys talk to each other? We're not really sure on this answer. Yeah, see that? Okay. Talk to each other, try to convince each other of your answer. Look back at your notes from last time. You can still change your answer.
This was wrong on the, lecture. Yeah, I got that. It's not the electron. It's like
What'd you say was wrong on lecture first?
well, it said this one on the lecture.
Was
quiz
right
that
or
it's
wrong?
Or something.
You think so?
I didn't do it.
All right, go ahead and send in your final answer there. It's like maybe one more person. You guys did a good job convincing each other for the most part. I'll show you again. Yeah, so most of you went towards D there. Okay, so there we go. Most of you went towards D, so flow of ions over membrane, that's it, right? Flow of charged atoms over a membrane, right? That's what it is. So that's what we're going to be talking about is pretty much we're changing the permeability of a membrane, two different ions, and then they flow across the membrane, and that's what electrical current is in the body. Okay. All right, so the first type of change like that we'll talk about is called graded potentials. Okay, so graded potentials are different than action potentials. Graded potentials are defined as this: a brief local change in membrane potential, meaning that it goes in the positive direction or the negative direction, just kind of briefly, and it doesn't spread, it just stays right in that area. Okay, and so we have two different types. It can either go in the more negative direction, which would be hyperpolarization, right? We're moving that away further down into the negative, or it can go up in the positive direction. That would be depolarization. Okay, all of these by definition, just a single graded potential will be below the threshold for an action potential. But we can do multiple graded potentials and they build on each other. And that's pretty much what that's showing here. If we get one stimulus there, it depolarizes a little bit. Okay, and then it starts falling back down, but before it can go down too far, we get our second stimulus, and then that can build on the next one. And so that's what this is referring to there. They can be summed, right? We can add multiple graded potentials together to get more stimulus. Okay, and this is how we start action potentials. If it's a depolarizing graded potential, or if we have a bunch of hyperpolarizing graded potentials, then that'll block action potentials because it moves us away from that threshold for an action potential. Okay, and so these spread slowly and they die out quickly. Okay, which is going to be very different than what we talk about with action potentials. Okay, so then action potentials are long distance signals, very fast moving. They don't die out. They just go all the way to the end of the neuron, no matter what. Okay. And so this is called the all or none principle, that once we reach an action potential threshold, it's going to shoot all the way down that neuron to the end. Okay. Also, there's no variation in how tall these are. As far as like how much depolarization we get, they're all the same height. Okay. So there's no variation in magnitude, and they don't add on top of each other. We just get a single one, and then it spreads. We can get another one and it spreads, but they don't add on top of each other. And there's no variation in their height. Okay, so those are two important definitions to think about. Okay, and so this is looking at that action potential graph again. Okay, and so we've already named these except for this one, but we named that one a while ago. Number one would be our resting state, right? Number two, again, would be what? What did we call that one last time? Depolarization. What did we call three? What was it? Repolarization. And then four was hyperpolarization. Okay, so this is the same states as before, the same phases. But then if we look down at this graph, it's telling us about changes in ion permeability in the membrane that are time aligned with the graph above, right? So right around the time that number 2 is happening, we see this big increase in the permeability of the membrane to sodium, right? Meaning that all of a sudden sodium can cross the membrane, okay? So that's going to be showing us what's causing number 2 is that sodium rushing across the membrane, right? And then right about here, we can see that potassium permeability increases a lot during repolarization. And we also see it's still pretty high during hyperpolarization, okay? So what causes an action potential is when we change the permeability of the membrane to ions. Really what that means is we're opening up channels in the membrane to allow for ions to cross. Okay, and what's going to dictate this is one of the channel types we talked about last time, which is these voltage gated ion channels. Right, so channels that open up or close depending on the voltage around them. Right, just like any protein, we can change its shape in different ways. We can change its shape by having something bind to it. We can change its shape by changing the temperature around it. We can change its shape by changing the voltage around it. And that's what's happening here, right? These proteins are designed in such a way that as soon as the voltage changes around them, they change shape and that causes them to either open or close. Okay, so we're going to be talking about 2 main voltage gated channels today. And so we have the sodium voltage gated ion channels and the potassium gated ion channels. Okay, so the potassium ones are over here. Okay, this is a potassium channel. Right, and you can see there that it really just has one gate. Okay, the big gate is right here. Okay, so potassium channels just have one gate. But then these ones are our sodium channels over here. And we can see those look a little bit different, right? We have this gate here, and then we also have this gate here. So those ones have 2 gates. Okay, 2 separate kind of doors that allow for changes on the membrane. See that? See, and then when we look at A there as the potassium gate or potassium channel, we can see it just has 2 states, which really is just open or closed. Okay, this one's closed right there. This one's open right there, right? We can see potassium rushing out. Here, no potassium can move. Right, so that's our potassium channel. But then on the sodium channel, it has 3 separate states. And you can see the names of them here, right? Here's resting state right there. Here's the activated state, which is really open. Right, and then inactivated state is a little bit different. Right, you can see this large gate is called the activation gate. And this smaller one is called the inactivation gate. There's kind of one that has a little round thing on the end. That one's called the inactivation gate. Then the larger kind of door-like one is called the activation gate. Okay, so it's a channel that has 2 gates, but it really has 3 states, right? So state number 1 is resting. State number 2 is activated. Sorry, state number 3 is inactivated. Okay, so sodium channel is the one that has 3 separate states. And that's going to play a really crucial functional role in the way that our neurons and our any electrically excitable cell works. Okay, questions on any of that so far? Okay, so very important that you remember potassium's got that one single gate, opened or closed, activated or inactivated. And then the sodium channel has 2 gates, an activation gate and inactivation gate. And the resting state looks like the activation gate is closed, right? And the inactivation gate is open. That's what resting state means. Activated state is they're both open. Inactivated state is the inactivation gate is closed, but the activation gate's open. Okay, so just remember that. And then what we're going to do is watch a brief video that kind of walks us through the steps of what's causing each of these different states in these gates. And then these are pretty much the steps there. And just like we've done before, I want you guys to kind of fill in the blanks on these slides, but we'll kind of walk through that together a little bit. Okay. But yeah, pretty much we'll watch the video and then you guys can, I'll kind of talk through this one for a sec right after the video. And then I'll have you guys fill in these few slides here. There's really 4 in a row that are kind of an activity to work through based on what we saw in the video. Okay. So let's see if we can get the video to work. Okay, so these are the steps that we kind of just walked through there. And what we'll talk about first is kind of just how this all begins, right? So when we're talking about a motor neuron, right? So here's a motor neuron right here, okay? But then we see another neuron, or maybe 2 different neurons coming and interacting with that motor neuron. We can see that the axons of those, what we would call like higher order neurons, are interacting with that motor neuron, and they're going to be starting the action potential on it. Okay, and so this would be what we'll get to this part in the class pretty soon. We talk about the different parts of the brain, but right in our frontal lobe, we have what's called the primary motor cortex, about a little strip of brain about right here. That's going to have neurons in it that go down to the spinal cord and then interact with a group of motor neurons for certain muscles, right? And so we would think of these axons coming from the primary motor cortex. Okay, and then this right here would be in the spinal cord, right? So those primary motor cortex neurons are coming all the way from the brain down the spinal cord, hitting a certain level of the spinal cord where we have motor neurons that are then going to send their axons out to the actual muscle cells. Okay, and so this is kind of like we're thinking about doing a muscle contraction, right? That activates a certain part of our brain, and then that sends a signal down to our spinal cord. Then we can contract that muscle. Okay, so the signal that's being sent right here is a bunch of these little graded potentials, right? So that's what we're going to get first. These are the graded potentials we were just talking about a second ago. You can see that they build on each other, right? And the whole point of that is to try to get to this threshold, which is -55, okay? If we can get enough graded potentials to get us up to -55, then you can see right there, it just shoots up super fast, right? So that's the threshold. Once we hit that threshold stimulus, then we're gonna get an action potential, okay? So what part of the neuron receives the stimulus to start an action potential? It can be the dendrites. But then you can also see that the soma, at least to see by that image, some of those axons are going directly to the soma as well, right? So they're interacting directly with it. We're going to create graded potentials out here. There's a bunch of little graded potentials. If we get enough of those graded potentials on this part here that we talked about last time, what was that first part? Oops, what was that first part of the axon called? Axon what? Hillock, right? So that's where the action potential begins, is on that axon hillock. And so that's where we're first going to see this big spike of an action potential. Okay, questions on that part so far? Okay, then here's what I want you to do. I want you to just kind of walk through. This slide is walking us through what's going on with the sodium channel. The second slide is what's going on with the potassium channel. Okay, the third slide is you're kind of having to put these in the right order. Okay, those images are not in the right order. Okay, but for each figure, I want you to state which ions are moving, the name of the stage, and then the correct order. And this is what I'll have you send in on your clickers first. Okay, so it's going to be the right order of those letters. Okay, so go ahead and work through those 3 slides, and then we'll come back together here in a couple minutes. So work through this with the people on your team. So really just saying open or closed on all of those.
Are you asking if they're open or closed?
That's it. So you gotta put open or close, yeah. So you can kind of do it based on these, oops, and these pictures too. Like where it shows, you know, you
Okay,
can see the state of those little gates.
okay.
Yeah. Mhm. So you can use those images on this slide and on the next slide to help you. Write it in there. See how you know this. Come on. You got it. Don't wait for me to uncover it.
What are we?
So just write an open or close. Yeah. Yeah. So you just write an open or close. You don't need to wait for me to tell you. If you make the decision yourself, you'll remember it better.
For the resting and then potential,
Mhm.
the inactive gate, is that also open during resting?
Inactivation gate is open during resting. Yep. Yes,
Okay.
if you go back to the states of the gates that we talked about right there. So you can see resting state, but that one's open. Right? And then inactivation, it's closed. Yeah.
One more question.
Yep.
For hyperpolarization, so
Uh-huh.
when the activation gate closes, because it relaxes, does then the inactive gate, is that still closed or does that release?
It's
as
gonna
soon as it's
reset back to its
Okay.
resting state, which, yeah, inactivation will be open,
Okay.
activation's closed, yeah.
Okay.
Mhm.
Yeah, one gate. Resting.
Just in the right order, yeah. So you're filling out these two slides here, and then with this slide, you're sending in the correct order of those letters. That's what you're sending in on your clickers.
But then what about this side? What's the difference?
Yeah, so they.
Got it all figured out, Jeff?
Yeah, we.
You didn't write anything down? That's what I'm telling you. You make the decision that helps you learn it. You just wait for me to tell you, you don't learn it, right? Goes one in ear, out the other. Just cross it out. You got room. It just says open or close. Yeah.
The activation and the inactivation.
The
Right.
gates are the states. So yeah, that's the inactivation gate. The big one is the activation gauge.
So when it's resting, it's closed.
Close and open, yeah. In order for sodium to get through, they have to both be open. So that's,
So would it both be open for depolarization? Or-
no, but we'll go over in a sec. For depolarization, yes. Yeah.
Okay, that makes
Mhm.
sense.
All right, so you're putting in all those letters in the right order, hitting send on your clickers. You got to hit send there.
That must be like, it must, I bet these first, and then some of them.
All right, make sure you hit the send button. About 20 or so more of you. Oh, about 20 more to go. Okay, make sure you hit send. Everybody go ahead and hit send to make sure you got them all in there. Still missing about 10. This is what the responses look like here. Word cloud format. Okay. I think most of you are in there. There's still about 4 that haven't sent it in. So maybe just hit send one more time. But let's go ahead and start in here. I'll leave it open in case you're still trying to hit send. Okay, let's start here with the sodium channel. Okay, so the sodium channel is the one that has the 2 different gates, right? We got the inactivation gate and the activation gate here. Okay, so during resting membrane potential, the activation gate is what? Open or closed? Closed. Yeah, good. Okay, and then what about the inactivation gate?
Open.
It's open, right? So that is back to this slide that showed the states, right? This is a resting state, right? That's what we're talking about. This one's open, this one's closed. Okay, so we got that one. Depolarization, in order for sodium to get through there, they both got to be open, right? So they're both open in that case. So the change going from the resting state to the activated state is just the opening of the activation gate, right? Inactivation gate was already open, so we're just opening up that activation gate. Okay. During repolarization, now the activation gate is what? It's still open, yeah. But what's blocking the sodium now? The inactivation gate, right? So it pretty much all happens at the same time. When we hit depolarization, the activation gate opens up quick, and then the inactivation gate is like slowly closing. In that matter of a millisecond or two, all the sodium rushes in, but then the inactivation gate kind of plugs it up right then. Okay. And then when we get to hyperpolarization, right? Activation gate is closed again, inactivation gate's open, and that allows us to be ready for the next action potential. I'll explain what I mean by that in a sec. I think my Apple Pencil is dying. It's not working too well lately. Okay, so that's our sodium channel. Okay, questions on that one? Okay. All right, so then let's go to our potassium channel. This one's easier because it's just opened or closed, right? So during resting membrane potential, is this opened or closed? Closed, okay. During depolarization, still closed. Repolarization, open. Hyperpolarization. Yeah, this one's a little bit trickier because during hyperpolarization, potassium's still leaving, right? That's what's causing it to become more negative. So it's kind of like in the early part of it, it's open, which is what it's allowing us to get down to the deepest negative potential. And then sometime around when it hits that very hyperpolarized state, then it closes again. Okay, so it's kind of open early and then closes in the later part of hyperpolarization. Okay, So then let's see what you guys said. So most people said ECABD. Okay, so there's two that look very similar, right? I heard some of you guys talking about that. Which two look somewhat similar to each other? E and D, right? Yeah, so some of you put D first, right? Which makes sense. They're both in the resting state, right? We see that the gates are, the activation gate is closed and the activation gate is open on both of those, right? So those are both in kind of a resting state. The only difference is this right here, right? That we have this local action potential hitting there first. So this would be number one, okay? You could also do it the other way. It doesn't really matter, right? You could say the other one's one and then the local action potential hits it, or local potential hits it. Doesn't really matter. Okay, we'll say this one is two here. Okay, and then what would be next? What's our next state, or what's our next phase? Sorry, that's not two. So some of you are wondering, wait, that doesn't make sense. That is not two. Sorry, that's gonna be the end point. Sorry about that. Okay, what's our first phase then that's gonna happen here? After resting, what do we do next? Depolarize, and which letter is that? Okay, so we got C. So we see all the sodium rushing in. So there's two, okay? And now what's next? What do we call it? After depolarization is repolarization, okay? And we see that right here, okay? So with two, which ion is flowing in which direction? Which ion?
Potassium.
This one right here, what's the ion there? That's our sodium, and which way is it going? Out or in? In, perfect. So that's the most important part you need to know. Depolarization is caused by sodium rushing in, okay? And then with number three, that's our repolarization. Which ion's moving there? Potassium, and which way is it going? Out, perfect. So we can call that potassium efflux, or it's just going out. We can call this sodium influx, or it's just going in, okay? And then what's the difference between A and B? Somebody tell me that. Potassium's moving in both of them, right?
Activation gate is still open.
There we go. This is the difference, right? So in B, that's going to be our number 4 there. That's going to be hyperpolarization. Potassium is still going out, but it's gone out enough to where we've reset the sodium channel, right? So this has been reset. That's what the resetting of that is caused by hyperpolarization, right? We have to get the membrane negative enough to cause that resetting, and that's what happens there. Okay, and then we can go to number 5 here, would just be like back to resting state. Okay. All right, questions on that? So take some time to kind of work through that, get that all in your brain. But let's go to this one as well. This is just kind of the matching of those parts of the graph there. Okay, so we're going to start with resting potential. Well, let's start here with E. Okay, so E is going to go, oops, sorry. E is going to be our first one that's kind of moving us towards the threshold, right? So we can see a little graded potential taking us towards the threshold. And then with depolarization is C. So we're going to say that's right there. We see the big depolarization happening, right? Now we're starting to repolarize in the next one. And so we'll call that one A, right? That was the beginning of repolarization. Okay, and then as we move down into hyperpolarization, we're going to reset the sodium channel. And so we're going to probably see that in B. And then this one's really kind of looking at the new resting state, which would be that one, D. Okay. Questions on that? And these are things you can kind of use to help you study along the way as well. Cover up those answers, and yeah. Okay, so let's go ahead and talk about this question for a second with the people on your team and take your best guess there as to what you think. Why does it matter so much? We got those 2 different gates inside the sodium channel. Great example of structure dictating the function of something. All right, go ahead and take a guess. Pretty much exactly split between C and D there. So maybe just debate those two answers with each other for a sec, and I'll give you a chance to change your answer if you want. Here's some very heated debate going on out there. I'm being sarcastic.
Haven't been talked about.
Yeah, I don't know.
All right, we got some little bit of movement here. Now we got 61% of you saying C and about 39% saying D. So this is our answer right here. And I haven't told you that yet. This is just kind of you putting things together. Okay. So we'll talk about why that's the answer in just a sec. Okay. So as an action potential is spreading down the membrane of a neuron, right? What's happening is we depolarize one part and then we depolarize the next part, but then the part right behind that is now in the repolarization phase. Okay. Which means it's in that inactivated state. Those sodium channels are stuck in that inactivated state. So it doesn't matter if we hit them with another depolarization, doesn't do anything. If we're stuck in that inactivated state, we can't reopen that gate. We have to first reset it via hyperpolarization, and then we can see another depolarization. Okay, so depolarization of neighboring membrane, open neighboring membrane, then we open the next voltage gated sodium channel. Okay, so what keeps it from going backwards towards the cell body? We have this thing called the absolute refractory period of an action potential, which means that when that sodium channel is in that inactivated state, meaning the inactivation gate's closed, activation gate's open, doesn't matter if we hit it with another depolarization, it doesn't do anything to it. It's in this absolute refractory period. Sodium channels have to be reset first, and then we can hit it with another one of those graded potentials, and then it'll reopen, we'll get the action potential again. But until we reset them, they're stuck in that state, and we can't have another action potential on those. And the next slide has kind of a little image that will help you understand that. Give you a second to finish writing that down though. Okay, so this little image kind of shows that. So if we're looking at one part of a neuron membrane, right? That's kind of what we're zooming in on here. We can see that there's some voltage gated sodium channels, right? Some potassium channels. So what happens is we depolarize the membrane up here first. Okay. That is then, so this is kind of shifting down in time, right? So the depolarization is spreading each step of the way. Okay. So now this part of the membrane is depolarized. Okay. And so then we're spreading to the next one down here. So now the depolarization part's here, but right behind it, sorry, my pen is not working. I need a new one. Yeah, but right behind it, you can see this is still in that inactivated state, right? See that? So right behind the depolarized part, it's repolarizing, it's in an inactivated state. It is frozen there. If we hit it with another depolarization, doesn't do anything to it, doesn't matter, right? So the action potential can't move backwards, right? This is electricity is just going to try to spread both directions. That's just what it does, but it hits that part of the membrane that it can't do anything to. So instead it just pushes forward. Okay. So now we're depolarizing this part, right? Behind it's repolarized. Same thing here. Now we depolarize this part. Right behind it is repolarizing. So we can't move it back. So the action potential is only going to spread in one direction, which is helpful in a couple ways. Number one is the brain wouldn't know what to do with the signal coming back to it on a motor neuron, right? What would that do? It wouldn't make sense. It wouldn't really do anything. There's nothing there to be released into the brain. And then secondly, it's really energy intensive to do action potentials. This uses lots of energy to get the membrane back to resting state. And so it'd just be a big waste of energy if we're just kind of letting electricity spread in both directions. So just this simple little thing, we got these two separate little gates, is what forces us to have afferent neurons and efferent neurons, right? Only signals traveling in one direction. Okay, questions on any of that? Okay. All right, well, let's talk about a few things surrounding action potentials. Number one is how fast signals travel down neurons, okay? There's two primary things that dictate how fast the signal travels. Number one is the width of the axon. Okay, so the bigger it is, the more sodium channels we can have. So then the more sodium can rush in all at once, right? We get a quicker depolarization. Okay, and then the larger it is, just like in the fire hose versus a garden hose, there's less resistance, right? There's more water on the inside. So there's more conductive material, and that electricity spreads a lot quicker. So that's number one. And then number two is a degree of myelination. Right, so that's that insulation that doesn't allow the electricity to spread outwards, stays inside, and that makes the signal kind of just skip from one unmyelinated spot to the next, like we talked about before. Okay, so our bigger neurons that are myelinated are going to be our fastest signals, and then the skinny ones that don't have myelin are going to be our slower signals. Okay, so in an individual nerve like this one, right? So we got a big nerve like this. This could be the ulnar nerve, sciatic nerve, something like that. We got all kinds of different size axons, right? See some little ones, some big ones, right? And so there's pretty much three groups of fiber types. Okay, there's group A fibers, which are very large and very fast, 150 meters per second. That is 334 miles per hour, right? So these are the neurons that carry signals to our skeletal muscle, to our joints, to our somatic sensory motor fibers. Okay, then we got our group B, which are kind of medium size. They have some myelination, not a lot. They go about 33 miles per hour. This is our autonomic nervous system and all the sensory coming from our vital organs. And then finally, our type C fibers are very small diameter. They don't have any myelination. They only travel a little over 2 miles per hour. These are our pain fibers and our very small touch fibers. Okay, one more clicker question here. Go ahead and take a stab at that one with the people on your team. Give you a minute or so to do that. Send something in when you're ready. All right, go ahead and just take a guess at that. This was a guessing one for sure, and that's reflected in the bar graph there. We're not sure. That's okay. So I'll leave it open there for a sec for a couple more of you to send it in. So the answer that's winning out the most is C. Okay. But really the answer that I'm looking for is this one, making it difficult to repolarize. Okay. Because what are we reliant on a potassium gradient for? Is for repolarization, right? So we normally have a lot of potassium on the inside, not much on the outside. If we do away with that by putting a bunch of potassium on the outside too, then we can't do repolarization, right? Because potassium is not going to move out anymore because it doesn't have a gradient to do so anymore, right? So if we can't repolarize, then we can't reset those sodium channels. And this is actually a life-threatening situation if we have high potassium, right? This is one of the key things that's measured in the hospital in everybody's blood is potassium levels. Because if you have high extracellular potassium, your heart stops, your diaphragm stops, everything stops because we can't send nerve signals anymore because all of our sodium channels get stuck in that inactivated state. Yeah.
I have a question. So, I tell people on my swim team that, hey, eat potassium for cramps.
Uh-huh.
Does that actually make sense? Or is
It makes
not make
sense. We don't really know for sure if that helps, but it does make sense because we do have some ion disruptions with muscle cramping, but it's probably more of a neurological fatigue than it is strictly with potassium for sure. But but it can help some people for sure. Yeah, definitely. Okay, we got a few minutes left. Let's talk about a few clinical applications related to this. So how many people have heard of lidocaine, Marcaine, things like that? Yeah, been injected with those maybe at one point in your life. Okay. What those things do is they block those voltage gated sodium channels. That's what they do. Those ones with the two gates we've just been talking about. That's what they do. They bind to those voltage gated sodium channels. They just get them stuck there, right? So now we can't use our sensory neurons anymore. We can't send that signal back to our brain that tells us something's wrong down there. So we don't feel the pain, right? It just numbs it. Okay. Epidurals are the same way, right? When during delivery. Okay. MS is related to the nervous system as well. Obviously, it's an autoimmune disorder that destroys myelin sheaths, specifically in the central nervous system. Okay, couple more clinical applications that are interesting. We kind of already walked through this one with potassium, but when extracellular potassium is too high, potassium won't exit as easily, so we can't completely repolarize. We can't reset the sodium gates. This happens during a spinal cord injury. When we damage some neurons in the spinal cord, they normally have lots of potassium inside of them, right? So when we break open some cells, we dump out a bunch of potassium into the extracellular space, which pretty much paralyzes temporarily all of the other neurons too, because they can't repolarize. Okay. And then part of the cocktail for lethal injection is a bunch of potassium, right? Like I said, it's a life-threatening situation if you have too high potassium. So part of it was that Cure drug as well that made it so you can't contract your diaphragm. But this is also part of it because it makes it so you can't send signals on your neurons. Okay, that's if too high of potassium. If we have too low of potassium, this actually drives our resting membrane potential lower. We're going to stay hyperpolarized because there's such a strong gradient, right? We have lots more leakage of potassium, which pushes us more negative. And it makes it harder to get up to that action potential threshold, right? If we're going deeper into hyperpolarization, we're further away from that threshold, and that can kind of cause paralysis, right? So this happens in elderly people or with lots and lots of vomiting. So, all right, guys, we're all done. Have a great rest of your day. We'll see you on Friday. Yeah,
Riding the bus?
I am. Yeah.
Okay.
Want to hang out?
Yeah.
All right.